Good morning, or I guess it's good noon for this video. So we've moved from the field into the lab in our freshwater ecosystems class. And the first thing we need to do when we get into the lab is, as a team, we need to inventory the number of samples we collected and brought into the lab to process, and also those that you may have collected in situ where it's direct data, and that will help you understand what you need to analyze. And usually by the time we get to this, you're about ready to write your draft field methods, and so this can help with that and clarifying in your mind what samples were collected in the field and which ones were in situ, so that you rack and stack that into your methods, your field methods section. So let's take a look at this overall table. I've divided it up into each of the teams, and then I've done a high level of the different types of samples we took in the field. So water, both filtered and unfiltered, chlorophyll A, and total suspended solids. For zooplankton, out at lake and freshwater tidal, we collected zooplankton, nowhere, not at the other sites. So we had two types of samples, the 44 micron samples into the buckets, and then the tow samples, the 202 micron. For benthic, we had grabs out at the deeper water bodies, and we had kicks at the stream. We didn't have any benthic for the wetlands. And then in the wetlands, we collected soil and vegetation samples. And then there were some other in situ measurements that we made, and so you want to have an accounting of those too, even though there's nothing you need to process in the lab for those. So let's start with lake. In the lake, the lake is 10 meters deep, and we collected water samples, zooplankton samples, the 44 micron ones, every 2 meters. And so that's 6 samples, one at the surface and then one every 2 meters. And so we should have 6 filtered samples. We should have 6 unfiltered samples. We also filtered for total suspended solids, so we should have 6 of those, and we filtered for chlorophyll A, so again 6. So for the lake, you should be looking for all of these filters and all of these filtered and unfiltered samples, and make sure that you have all of the labeled samples with the proper depths on them. Make sure that you don't have any unlabeled or confusing ones. Not that you can do a whole lot about it, but at least you know what you're missing. So for zooplankton for the lake, we also collected 6 44 micron samples. And then for the tow, we did vertical tow from the epilymnium up and then a full column. And so we've got 2 of those. Epi, and I'll say FC for full column. So these are every 2 meters, okay? And same here, every 2 meters. Then for benthic, we collect 2 grabs at the lake. So 2 grabs. We don't have any soil or vegetation from the lake, but then in situ measurements were our YSI measurements, and we should have one of those every meter, so 10 or 11, depending on how deep we're able to get there. We also collected light and sec. So we did an alkalinity titration test out at the lake and also at all the other sites, and so we have alkalinity data that can be used to determine total dissolved inorganic carbon, which is a problem in problem set 3. Okay, so let's go to stream, just for contrast. So at the streams, we went to 2 different streams, and we collected 2 samples at each streams for a total of 4 samples. So what I'm going to put here is 4 filtered, 4 unfiltered, and then 4 and 4 chlorophyll A and TSS, and this is 2 per stream. So when Stream Group does chemistry or processing of those filters, they need to process all of these, so they need to check for the number of filters and water samples, filtered and unfiltered, that they have. So zooplankton, no zooplankton at the stream, but benthic was huge, so we have 4 kicks, 2 per stream. There's no soil or vegetation at the stream, but we did have, and I'm going to add this because I forgot it, stream morphology, which is really, it was a measurement, but it's something that we measured directly at the stream, and so you're going to have that data on a data sheet. So what did we get? We had depth that was measured across the stream and that you'll take an average of, and then we also had velocity measurements for each stream. So this is something, because it's data that's sort of in situ since we measured it and just wrote the values down, you need to double check that. I'll put 2 sets. When you put the data sheet together, this is actually something you need to verify that you have it. But you have this, and you're going to do some calculations to determine discharge. You'll also create a graph that shows the stream morphology. Okay? Other in situ measurements that we did at the stream, we did YSI, 2 measurements per stream, so for a total of 4 measurements. We did not do light because light requires some depth. We did not do Secchi. Same with Secchi. We need to have at least a meter to determine Secchi, and it's really not something that you measure in streams. And we did do an alkalinity test. So that's what we have for the streams. I want to say freshwater tidal. Basically, you can replace each of the water and zooplankton samples with 2. So, put that here. Oops. 2, and that's per site. So you have a total of 4 total here. Water, and it's the same for the zooplankton, or that you're going to have 2 per site, and for the toes you have 2 horizontal toes, so 2 HTs. So you should have a total of 6. For the grabs, again, you have 2 per site. And remember that we had an embayment, and we also had the main stem of the Potomac River. So those are your 2 sites. So no soil or vegetation, no additional unique information. And again, in situ would be just like the lake, and where you measured each meter down for YSI. You also have light, Secchi, and alkalinity. So I'm going to just draw a little arrow here. Same. Okay, and then finally for the wetland, you have a single stream here, and so we just take 2 water samples. I'm going to put a big 2. And we do all of the tests that we do at every site, except you only have the 1 stream, so we take 2 samples. No zooplankton, just like the other streams, we aren't collecting any zooplankton. No benthic either. I don't know if you remember from the previous video, but the stream that we're sampling water in tends to be very degraded, and so we haven't had much luck in our attempts to find any benthics, and so we don't waste your time collecting any benthic from that stream. But real biggie for the wetland is the soil and vegetation. And so for vegetation, put a V. We had 2 transects, 30 meters. We took 15 samples per transect for a total of 30 samples. So for vegetation, we have the percent cover and ID. So I'm just going to put PC for percent cover, and we've got 15 of those. For the biomass, we take a sample every other sampling location because it's a lot of biomass. And so, I'm sorry, this should be 30. 1 for each transect. And for the biomass, we'll have half the number of samples, 15. And then for the soil, let's do soil. We took a soil sample every location. So each one of our sampling locations, so we're going to have 30 actual soil samples that we're going to use for soil organic matter, saturation or moisture, and help me.
Redox potential?
Yeah, well, pH. Okay, so then we have some in situ measurements. And these included redox potential, which I'm calling EH, and also we had saturation and rough pH. Okay, and then we also had YSI and alkalinity, just like the stream, but just 2 of those. So 4 streams. So you'll have access to all the data for all of these sites. And one really interesting assessment that the wetlands group can do, since they don't have very much in terms of water data, is actually consider their site a highly degraded site and the stream sites more pristine, and then they can compare the 2 when it comes to all the different data collected. Okay, so each team should, after determining what they should be able to find, they should go and find the samples and make sure that they keep a running inventory of what they actually process and what they've done calculations on.